Hello and welcome to Amira Software product demonstration webinar. So from my side, today we will revisit the three use cases which have been introduced during the introductory webinar already. Our goal is to show you how Amira Software supports researchers to understand the bone structure and physiology. So today we will talk and demonstrate the separation of the trabecular and cortical bone segments in microcity data. We will talk about the characterization of the growth plates in bright field data, but we will also show you how full field displacement analysis of a bone upon deformation experiments can be performed, and this will demonstrate on microcity data. But before Rosa starts with the actual demonstration of the product, let me briefly recall the individual use cases. So understanding the structure of the trabecular bone is essential for investigating its physiological function and its role in different diseases. Here we show an example of the epiphysis of the murine tibia, which has been imaged by microcity. Due to the similarity in bone signal, the segmentation of the trabecular bone separately from the cortical bone is often performed manually, which is highly time consuming and also prone to errors. In the following demonstration, you will see now how the cortical and trabecular bone segments can be segmented separately. In addition, also the bone marrow space in a semi-automated way, which provides a very time-saving, efficient, and reliable method.
In this use case, we are going to show you how semi-automatic tools, computational modules, and recipe allow to easily accelerate the segmentation of marine tibial epiphysis and how to segment in particular the trabecular bones. After the segmentation and characterization of different structures, 3D surfaces can be generated and quantitative analysis of morphometric data can be also collected. The data and recipe are kindly provided by Eva Herbst from the University of Zurich. So here we can see an image of the Amira project, where in the project view we can observe the workflow, and in the 3D viewer we can observe the grayscale image of the cortical and trabecular bone, and the 3D surface of the trabecular and marrow space. So now I'm going to switch in Amira Software to show you more details about this project. Okay, so here we are in Amira Software, where we can observe the grayscale image of the murine tibial bone, and specifically we can observe the epiphysis region and the metaphysis region. And then there is a 3D reconstruction of the trabecular and marrow space, which correspond to the epiphysis region. So now I'm going to switch to a single viewer, where basically we can move with the mouse the slices like this, or we can also set up a specific ortho slice using the slice number. Moving, so basically the ortho slice allow you to observe the dataset to a different cross sections. So now I'm going to switch off two ortho slices and to keep just one. And then again, the top structure corresponds to the epiphysis, and the bottom side of the structure, this is the metaphysis basically. So we are going to focus on the segmentation of the epiphysis. So in Amira, I want to show you that we were able to segment the epiphysis. So here you can observe in green the segmentation of the epiphysis structure. And then from that we were able to segment the marrow space of the epiphysis. And finally we segment the trabecular structure. So how we can basically do this in Amira? So we have a specific segmentation plus workroom, which allow to use different semi-automatic tools to segment specific structures. And also a recipe was created by Eva Herbst to segment the Dravecross structures. So now I'm going to switch to another project to see more details about the segmentation part. Okay, so here we are in another project where you can see the original dataset. We applied the filter to better improve the quality of the grayscale image. So now we switch to the segmentation plus workroom. We select the filtered data, and we are going to basically create a new label. So first of all, we are going to focus on the segmentation only of the epiphysis. So that's why we can rename the material 1 as epiphysis. And then we want to isolate basically the epiphysis from the rest of the metaphysis structure. So the second material will be out material, because we want to include the dark space and the metaphysis region. We are going to use the watershed, which basically create a gradient of the selection. But first of all, we need to create the seeds for the watershed, and we can use the brush tool. Then we can work using the 4 viewers to better see the selection, or we can also change it to a single viewer like this. So first I'm going to select the structure which corresponds to the for the epiphysis material. So now I will add the selection to the epiphysis material, and then I'm going to select the rest of the structures, the dark region, the metaphysis. And these will be included in the out material. So now we can press on watershed, create a new gradient image, and then we can press on compute label. So now we can observe that after a few seconds we obtain the result. So we have the segmentation of the epiphysis and the segmentation of the background around the epiphysis. So we can also observe that there are some specific structures that probably not belong to the epiphysis region. So that's why we can use, for example, the lasso tool to manually select these regions. And then we can, for example, add this material to the out material. And then automatically they are removed from the epiphysis material. So now we want to focus on the segmentation of the marrow space of the epiphysis region. And we can use basically the mask created by the out material. So what we can do is to create a selection from that material and to create a new label. And in this new label, we are going to add the out material. So we can rename the material 1 as out. And we can also change the color using again the pink color. Okay, so now we want to focus, as I said, in the marrow space, and we can use the thresholding tool. So the thresholding creates a selection from the current masking setting. We need to adjust the range in order to select the marrow space region. But if I switch off the 2D of the auto space, the auto material, you can see that the thresholding also considers the dark space of the auto material. But we are going to lock that material, so the out material. And now we can create a new material, rename as a marrow space. We can also change the color, for example, with the yellow one. And now we can create a selection and add this selection to the marrow space material. So as you can see, the marrow space is there, so it's segmented too. We can now basically delete the out material because we don't need it anymore and switch off the thresholding tool. So here we have the marrow space. If we go back to the previous label file, we can also delete the out material. And now we can just keep the epiphysis. So now if we go back to the project, we can see the two files that were generated after segmentation. And in the last file, you can see the segmentation which corresponds to the marrow space. So now we can create a recipe because we want to segment the trabecular structure. And we want to use this recipe that was created by Eva Herbst to segment the trabecula. Our input data is now the last file, and now we can press on apply. So the recipe is in particular a collection of some morphological modules. And now we can link the color wash to the final results of the recipe. So here you can see the segmentation of the trabecula. In the recipe player, you have two different options, open and export. If I click on open, I can see basically the recipe, the list of the modules that were created in order to segment the trabecular structures. So based on these results, we were able to basically segment the different structures, so the trabecula and the marrow space, as well as the epiphysis. And now I'm going to show you that in Amira, based on the segmented file, you can generate surface using the generate surface module. And here you can see the 3D surface of the trabecular structure bone and the marrow space. You can also quantify some morphometric data. For example, here we use the surface area volume module, and we were able to calculate the area and the volume, in this case, correspond to the trabecular structures. So finally, in this use case, I showed that it's possible to segment the different structures of the bone using semi-automatic tools available in Amira software. We also showed that it's possible to generate 3D surfaces and also to quantify morphometric data, for example, the area and the volume.
As the second use case, we would like to present to you data which was provided by the company Inoska. Here, the researchers were interested in segmentation and analysis of the proliferative and hypertrophic parts of the growth plate of the bone. And these they wanted to use for better understanding of the mechanisms which are related to certain diseases. In the following demonstration, you will see how to perform a reliable automatic separation and quantitative characterization of those two different segments. And for this automated workflow, including pre-processing and segmentation steps, will be used.
In this use case, we are going to show you how to characterize the growth plate of the bone using automatic segmentation and quantification tools available in Amira software. The data are kindly provided by the InnoScale company. First of all, here, this is the complete Amira project. The two images in the 3D viewer shows the two kind of cell population. So we have the grayscale image and also the label image. So the hypertrophic cells are in magenta and the proliferative cells in green. Then the computational module called the sieve analysis allows to easily separate the two cell populations. Quantitative data can be also extracted in Amira. So let's go in Amira software and then I can show you more details about this project. So here, this is the project. This is the grayscale image of the original dataset. So we can observe the hypertrophic cells and the proliferative cells. And what we want to do is basically to separate the two subpopulations and to characterize and analyze them. So in order to do that, the product expert created a different recipe. And first of all, the module, which is called the major recipe player here, it was used to run the first recipe. So what we need to do is basically to upload in file name the recipe, which is chondrocyte step 1 in this case. And then we press on open. And then we press on apply. So now I want to link the ortho slice to the results, which is the processed one module. And basically this recipe allows to resample the region where there are the two cell population into a convex shape. And then the extract-oriented labels resample this region along the main axis, which is the vertical one. So now the resampled information was included in the grayscale image, as you can see now here. And now we can proceed with the next step, which is basically the separation of the hypertrophic and the proliferative cells. So another recipe was created, and it was run in the image recipe player, the number 2. So the second recipe, together with the sieve analysis, allowed to really separate the hypertrophic from the proliferative cells. So now I want to show you more details about the second recipe. So that's why we go in the image recipe designer here. I upload the recipe. Which is, in this case, the chondrocyte step 2. And then I import the data, which is the resampled one. So now we see in the input and output the grayscale image. We are in the step 1 right now. So the recipe is constituted of 15 different steps. And in particular, I want to show you the step number 11. Because it's able to basically convert an image into a label field. If I zoom into the cells, we can observe that the cells are divided in different colored lines, and these are repeated in the horizontal way into the other cells. We can extract quantitative information related to the length of each line with label analysis. So here we can see the length of each lines, which basically correspond to measure the total thickness of the hypertrophic and proliferative cells. And this is repeated for all lines. Then a labeling module was used to label the different cells, and it was quantified the barycenter of each cell. The quantitative information of the barycenter was then used to proceed with the sieve analysis. So now if we go back to the project here, I can show you the results obtained after the sieve analysis. The sieve analysis is based on the barycenter, as you can see here in measure, and the cells are read as bins. If now I link the color wash to the sieved results, we can observe the separation between hypertrophic and the proliferative cells. And if I move the red lines here slightly, we can adjust the separation between hypertrophic and the proliferative cells. So now if we want to proceed with the quantification of the two cell population, we are going to use a third recipe. And this time the recipe is the chondrocyte step 3, which is here. And the input data is the sieved results, and the data correspond to the last results obtained from the second recipe. And here we can observe 4 different results. So the processed 0 basically correspond to the length measured for the proliferative cells. And then the processed 1 shows basically the density of the proliferative cells, which in this case is around 25%. And then the same kind of analysis was carried out for the hypertrophic cells. So we have the processed 2 result, shows the length measured for the hypertrophic cells. And then the processed 3 shows the density of the hypertrophic cells, which is around 40%, as you can see here in the table. So then in Amira, we can also create histogram using the module histogram, and we can also show in the plot the histogram. Finally, in this use case, we were able to resample to show how it's possible to resample an image in Amira software, how it's possible to separate two different kinds of cell populations, and how to perform a quantitative analysis to characterize the two cell populations.
So in the last use case, we would like to show you the capabilities of Amira software for analysis of the mechanical properties of the bone. To investigate mechanical properties, bone samples are typically imaged before and after application of a well-defined force, and the structural displacement is then calculated. From this calculation, one can derive then different mechanical properties, for example, strain. But the major challenge is to do these calculations in the entire specimen at very high resolution and precision. And in the following, we will demonstrate how this analysis can be performed in Amira software.
In this demo, we are going to perform a digital volume correlation analysis and visualize the corresponding displacement and strain fields. The bone cement data used in this demo is a courtesy of Gianluca Tozzi at the University of Portsmouth. So here we can see the complete Amira project. In the 3D viewer, we have two grayscale image of the bone cement data before and after the compression. We cannot really see too much differences between the two data. So now let's look on the structure. So we have basically the dark gray structure is the cement, and the light gray structure corresponds to the trabecula, and then the dark area corresponds to the hair or soft tissue. Then there is a volume rendering of the bone cement, which shows the displacement vector from the original acquisition, so before the compression, to the compressed data. And finally, there is also an image of the 3D surface of the axial strain in the bone face. So let's go in Amira software, and then I can show you more details about this project. So here we are in Amira software. So first of all, we need to open the data, the reference data and the pick and force data, which are stored in the tutorials and are available in Thermo Scientific Amira Viso product. So I'm going to open the data. And first of all, I can link an orthoslice to the reference data. And I can also link an orthoslice to the compressed data. So we cannot really see too much differences between the two data, but if I start to switch on off one of them, you can really see the difference. So because some regions are compressed. So before to proceed to perform the digital volume correlation analysis, we need to generate a finity structure where we want to perform the analysis. So that's why we're going to generate a global DVC mesh. And now here we have the results. So to visualize the DVC mesh, we can link a grid view. And we can also adjust some parameters to better visualize the grid view. So we can change the color to the yellow one and also the draw style with the wireframe. So this is the finity structure where we are going to perform the digital volume correlation analysis. So now we have all elements that we need to perform the analysis. So we have first the reference data, the compressed data, and the results obtained of the DVC mesh. So now we can basically link the computational module, so the DVC global approach. And the reference volume is the reference data, the deformed volume is the pick and force, and the reference mesh is the last results of the DVC mesh. So now I press on apply, and the software will perform the analysis and will generate different results. But in particular, we are going to focus on the local displacement result and the mechanical strain. So for the local displacement, we can use the vector field to visualize the displacement. But first of all, we are going to perform at the magnitude. So now we have the results of the magnitude, and we can link the vector field to the local displacement. And in the color field, we can link the results of the magnitude. And we can also change the coloring. And if we go in the magnitude, we can also change the color map with the physics. So here we can also link the data legend, which map basically to a magnitude. And now this is the vector field. We can also a bit change the thickness of the vector field. And finally, the vector shows the displacement from the original dataset to the compressed one. And then now we can also export components that belong to the mechanical strain, and we can also convert the components into a volume. So that's why I'm going to open another Mira project, and then I can show you more details about the mechanical strain and how you can generate the surface. So here we are in the project where we can see that this is the same project. So we performed the digital volume correlation analysis and the magnitude. But in additionally, we also use the extract scalar components module to extract information from the mechanical strain. So basically the components, the component in Z. So here are the results. And then we converted the scalar components into a volume. So here we generated a corresponding volume for the mechanical strain. And then we generated a 3D surface, which is based on a label file. And we link the information, the volume of the mechanical strain into the 3D surface. So basically here you can see the visualization of the axial strain in the bone face. So finally, in this demo, we showed how it's possible to perform a digital volume correlation analysis in Amira software, how it's possible to observe and analyze the data, and how it's also possible to convert the components, the scalar components, into a volume, and how you can also visualize the information using a 3D surfaces.
So I think we have seen that Amira software increases the time efficiency and provides reliable and reproducible results using automated and performant tools for the analysis of bone and tissue specimens. But I would also like to mention that the software provides tools for all the steps you require for quantification of your image data, starting with data import, then going for data pre-processing or denoising. Of course, high quality visualization, where you also can explore your data in 3D or 4D. This is, of course, in most cases, followed by segmentation, which is the major step of image analysis, where you can clearly identify the objects or structures of your interest. After segmentation, you can perform measurements and quantitative analysis within the Amira software. Also, you can plot or do statistics on the data you get. And last but not least, you can create movies or screenshots for your presentations or publications. So I think with this, we can sum up that we are able to demonstrate how powerful Amira software is for bone and tissue research. And now it's time for the Q&A session.